Hello and welcome to Life at PALS. Today we will go over a biology, IGCSE Biology paper 2, so for the extended syllabus. This is the specimen paper from 2023, so it is up to date with the new syllabus for the 2023 examination. And yeah, let's get started. So first question is about plants and light, as we can see. And the plant growing towards the light. Which characteristic of living organisms does this show? This is sensitivity. The plant responds to the light, it senses the light, can detect the light, and it responds to it, so this is sensitivity. Lichens are formed from two different organisms living together. Organism X and organism Y are found in most lichen. The table shows some of the characteristics of organisms X and organisms Y. Made of strands called hyphae. Hyphae have cell walls and many nuclei. Y single cell contains nucleus and chloroplasts. So yeah, we need to know what hyphae are. That's a bit tricky. This is a structure in fungi. So that's just something we need to know. It's like the mycelium of fungi, the hair-like structures. These hair-like structures is what we call hyphae. So yeah, X is a fungus, and since Y is single-celled and has chloroplasts, it must be a protoctist. Protoctists, 5 kingdoms, are animals, plants, fungi. These are the multicellular eukaryotes, multicellular organisms that have cells with a nucleus and organelles. And then we have a 4th kingdom, the protoctists. These are eukaryotes as well, so their cells have organelles and a nucleus too, but they are single-celled. So therefore answer B is correct. Which part of a plant cell controls the movement of substances into or out of the cell? This one is a bit misleading. We don't need the word plant here at all. So what part of a cell controls the movement of substances into and out of it? It's the same in all cells. It's the cell membrane. By the way, if you want to answer the questions by yourself, if you want to try yourself, you can pause the video after, or for every question, attempt it yourself and then continue with the video. We will go through this relatively quickly today. The diagram shows the structure of a plant cell. We can see this hair-like extension here. So what is a function of this specialized cell? It's a root hair cell, and therefore it's part of the roots. And what do roots do? They absorb stuff, so it's either A or B. It absorbs carbon dioxide from the air. No, it's underground, so it must absorb ions from the soil, and it does that together with water from the soil. How do carbon dioxide and oxygen move into and out of mesophyll cells? Since they are very simple molecules, which are non-polar and very small, these molecules can diffuse across cell membranes, so therefore diffusion. These are the major 2 molecules that you should remember in terms of being able to diffuse in and out of cells. 4 fleshy cut potato cylinders were soaked for 1 hour in different salt solutions. The potato cylinders were then pinned to cork blocks. 2 of the potato cylinders are shown. Which solution would cause the potato cylinder to be the most flaccid? So here we see the word flaccid. Turgid is the opposite. Even more extreme than flaccid is plasmolysed. And yeah, this is obviously caused by osmosis. So what has happened to our potato cylinder is that water has moved out of the cells, out of the potato cells, and therefore it all becomes less stable and it gets a bit floppy and it hangs down. So for this to happen, we need a higher concentration of dissolved substances, of solutes in the surrounding solution around the potato cells, because high concentration of solutes means low water potential, and water then moves from high water potential to low water potential out of the potato cells. So therefore we probably pick the highest concentration of salt solution, 1 mol per deciliter cube. Highest concentration of salt, one more time, we'll have the lowest water potential, so water moves out of the potato cells by osmosis. Number 7, the data shown. The data show the concentrations of sugar and starch in an onion. No starch and some sugar. The onion is tested with Benedict solution and iodine solution. Which set of results is correct? So there's no starch. Iodine solution naturally is brown, and when starch is present it goes blue-black. So therefore our iodine solution must be brown because there's no starch present. So it's not A, it's not C. And Benedict solution goes brick red when sugars are present. So therefore it must be D. Which statement about the structure of DNA is correct? We know our DNA has 4 different bases, A, T, G, and C, and we should know that A always pairs with T and G always pairs with C. So that means B is correct. A pairs with T. The diagrams shown are protease enzymes catalyzing the breaking of part of a protein molecule into smaller pieces. Which diagram has the correct labels? So the reactant in enzyme reactions is called substrate. So substrate is correct here and here. B is definitely wrong. This is not the product. The active site is this inner bit. So therefore the products are these things here, what are produced. So this is not the product, therefore this is wrong. So active site here and here is correct. Okay, substrate is wrong here. So this is the enzyme, this is the active site, this is the substrate. C is correct. A student wrote some notes about enzymes. She wrote the shape of an enzyme or active site of an enzyme is to an area on the substrate. This area on the substrate can fit into it to form an enzyme substrate complex. So when enzyme and substrate stick together, it is an enzyme substrate complex. So it is not B or C. And the shape of enzyme and substrate are not similar. Now you can see the substrate looks very different from the enzyme. But what is correct is that the substrate fits into this structure here, which is the active site, as we already said, and these two structures or these two shapes are complementary to each other, like a key and a lock. So therefore it must be A. Now the active site of an enzyme is complementary to an area on the substrate. 11. The apparatus shown was used in an experiment. The carbon dioxide content of the water in each test tube was measured at the start of the experiment and again 3 hours later. In which test tube will the carbon dioxide concentration decrease? So first of all, we know that plants, when they are able to photosynthesize, they will absorb CO2, which means in the water it would go down and they would release oxygen, so it would go up in the water. Animals And as well, plants in the dark respire, and therefore they release carbon dioxide to the surrounding, and they would absorb oxygen. So the only thing that makes sense here is C. We have a plant that receives light. There's no animal in our test tube, and that is the only situation where carbon dioxide will decrease because photosynthesis is happening more than cellular respiration. In all other cases, there is more cellular respiration happening, releasing carbon dioxide and increasing the concentration. 12, the diagram shows a cross-section of part of a leaf. What is the name of the cell labeled X? So we need to know the structures of a leaf. X is a palisade mesophyll cell. These cells here are the spongy mesophyll cells. Here we have the upper epidermis with the cuticle on top. Now we have air spaces in between the spongy mesophyll cells. Down here we have the lower epidermis, and this is a guard cell forming a stoma opening. So the structures in a leaf. Why do plants need nitrate ions? Plants can make their own carbohydrates from photosynthesis. They can change these carbohydrates into many, many other substances, such as lipids, for example. Why is that possible? Because carbohydrates and lipids both contain the elements carbon, hydrogen, and oxygen. They cannot change carbohydrates into proteins because proteins additionally contain nitrogen. So that means that they need their nitrogen for amino acids to build proteins. Now, amino acids are the building blocks of proteins. In which part of the body of a mammal does physical digestion occur? Physical, another word for mechanical, so breaking food down into smaller pieces without chemical change. That's happening in our mouth when we chew the food. The diagram shows part of the human digestive system. Where is protein digested by trypsin? Trypsin is an enzyme that is produced in the pancreas and released into the small intestine, specifically the first part of the small intestine, the duodenum, and that is structure A. So it is produced in D, but it is released into the duodenum, small intestine, and that's where it's active. The photomicrograph shows a cross-section through the root of a buttercup plant. So we have this inner structure here, which looks a bit like an X or fidget spinner or something like this. And then we have some vessels here as well. Again, we need to know which structures are where. Usually the xylem is towards the center, and the phloem is more on the outside, and this is the same here. So this part is the phloem, and this part is the xylem. So X is the xylem. What is the function of the xylem? The xylem transports water and dissolved mineral ions from the roots through the stem to the leaves. Sugar is transported by the phloem. Roots and leaves both act as sources and sinks for sucrose and amino acids. So a source provides the sucrose, it's where it comes from. A sink is where it is used, or where it's needed. Source and sink, going from here to here. And amino acids, roots and leaves, both act as sources and sinks for sucrose and amino acids at different times of the year. At which point in the year are the roots most active as a source? That is probably during the springtime, when new leaves start to grow, because that is when the leaves are not doing photosynthesis yet, but a lot of energy is needed for them to be able to grow. No, they do a lot of mitosis, they make new leaf cells, all of that requires energy, and this energy is provided from storages in the root, which in this case serves as a source. Number 18, the diagram shows a section through a heart, which blood vessel is the pulmonary vein. So a vein is a blood vessel that goes into the heart. We know that blood comes into the heart into the atria, and then moves through the heart and out of the heart through, is pumped out of the heart by the ventricles through these blood vessels here. So that means it's either D or A. A, the right side of the heart, receives deoxygenated blood from the body, and that is the vena cava. So A is the vena cava, and B is the pulmonary artery, which pumps the blood to the lungs, and that gets reoxygenated, and oxygenated blood then comes into the left atrium through the pulmonary vein. So the correct answer here is D. And then it will be pumped out through C, which is the aorta, to the rest of the body. The thickest and strongest artery in our body, the aorta. What happens to the heart valves when the ventricles contract? So when the ventricles contract, and we still have the picture here, that means this space gets compressed and the blood inside is getting pressed together, it gets high pressure, which causes the blood, let's actually clear this up a little bit. So when the muscle contracts, then the blood is pushed out of the heart only through the aorta. Here this is a valve, and we can see that when blood pushes against the valve like this, it will push the valve, the flaps of the valve together, and it will cause the valve to close. So there's no blood being able to flow back into the atrium. So that means this valve closes. What is this valve called? This is an atrioventricular valve. Pretty straightforward name, very long but straightforward. It is between the atrium and the ventricle of the heart. Now here the atrium and the ventricle. The atrioventricular valve is between these two chambers. So this will close, so it's either A or B, and this valve here separating the ventricle from the aorta, you can see it has another orientation. So when the blood pushes against it like this, this valve will open up, so the semilunar valve will be open. So the correct answer is B. What are the approximate percentages of oxygen and carbon dioxide in inspired air that we breathe in? There's almost no carbon dioxide in the air, so that leaves us with only 0.04 as an option, and 20% more or less matches, so that is C. Expired air contains around 4%, 5% carbon dioxide. So 0.04%, that is a number you should know, is for carbon dioxide in the atmosphere and in the air around us that we breathe in. The diagram with the structure labeled X shows a bacterium with the proteins on its surface. The diagram labeled Y shows proteins made by the human body. So proteins on the surface of a bacterium, which are used by our body to identify the bacterium and other cells of these proteins as well. These are called antigens. They're a little bit like a fingerprint that that's used for identification, that's unique to this bacterium. So X is an antigen, therefore we can exclude these two. Now our body produces, if it triggers an immune response, this bacterium, our lymphocytes, our B cells, will produce antibodies, which is Y, that are complementary in shape to the antigens. So they fit onto the antigens, and the structure that fits on them is this. Again, like enzymes and active sites and substrates, a complementary shape, lock and key. So therefore it must be A, antigen, antibody, and the correct shape. Which environmental conditions must always be present for seed germination? So, very common mistake in this question is to say light. Now, plants need light, yes, but germinating seeds do not need light. Why don't they need light to germinate? Well, seeds are usually under the soil, and underground there is no light, even if it's very bright on top of the soil. If the seed is too far down, it will not receive any light. So that's why light is not required for seed germination. And yeah, plus the seed itself cannot do photosynthesis. So what the seed needs, it needs some oxygen to be able to do respiration, because it has food storages. It needs to break them down, but it doesn't need carbon dioxide. So it's not B, it's not C. No, it cannot do photosynthesis yet, so it doesn't need carbon dioxide. So that means A can be excluded too. Water and oxygen, yes. No? So oxygen for respiration, water to get absorbed by the dried out cells so that the cells can start working again. Water very important for all living things. So D is correct. The diagram shows a reflex arc. Which structure is the sensory neuron? The sensory neuron is what senses, what detects a stimulus. So we can see the hand touches the hot pan, and we can see here nerve endings. So that is probably the sensory neuron, which must be A. Therefore, we can see the sensory neuron goes into the spinal cord, where it's connected to B. A relay neuron passes the information on, and to the motor neuron, which is C, which carries the impulse to a muscle. So if it's a bit difficult to see like where it comes from, how else can we know that A is a sensory neuron? A sensory neuron is the only type of neuron that has its cell body on the side of the axon splitting off, and the axon going in both directions. We can see that in a relay neuron, the cell body is at the end, and we can see in a motor neuron, the cell body is at an end as well. So only sensory neurons have their cell body on the side branching off of the axon. The axon is the long part of the neuron, and we can as well see that these connections is a synapse. Synapse only work unidirectional, so information is passed from this part here onto the cell body, and therefore we can see that the reflex arc must be in this direction, and it cannot be in this direction. And we know that a reflex arc always starts with a sensory neuron, so that would be the third way to figure out that A is the sensory neuron. What happens to the nucleus in the iris? What happens to the muscles in the iris to make the pupil smaller? So the iris is the structure around the pupil. The pupil is the opening. So if this is the pupil, then the iris is the colorful part in our eye around this structure. So our iris has ring muscles, which are called circular muscles, and it has muscles that work as antagonists to these ring muscles, and these are called radial muscles. So when ring muscles contract, then the rings get shorter, which means the iris will get smaller, and this causes the radial muscles to get stretched out, so they will relax. The opposite is true. When the radial muscles contract, they will get shorter, and this causes the ring muscles, the circular muscles, to get wider, and the pupil will get larger. So that means that to make the iris, to make the pupil smaller, all of this has to move inside. That means our ring muscles, our circular muscles contract, and the radial muscles relax. B. 25, a student used two seedlings, X and Y, to investigate phototropism. The diagram shows their investigation. So this one looks a bit tricky. Let's see. Which statement explains the difference in the results between X and Y? So here we have a shoot, first of all. Phototropism as well is the response of a plant to either grow towards or away from the light. So usually shoots grow towards the light. How do they do that? This involves the plant hormone auxin. Now in a shoot, a high concentration of auxin, so if auxin is high in a shoot, if auxin is high, then this causes cell elongation. So if we have a shoot like this, a normal shoot, without putting any piece of glass into the shoot, just a normal shoot, and we shine light from this side, what will happen is this causes auxin to get more concentrated on the shady side of the shoot, so you will have a higher auxin concentration. This means that the cells on the shaded side will elongate more when the shoot grows than the cells on the bright side, and this causes the whole shoot to bend towards the light. So we can see this works in Y because here the auxin is able to spread towards the cells on the shaded side, causing cell elongation, and our shoot bends towards the light. In experiment X, up here this does not happen because the piece of glass prevents the auxin, which is produced, this is actually something I should have mentioned, auxin is only produced in the tip of shoots. So only here in the tip of the shoot, this is where auxin is produced. So the auxin that's produced in the tip of the shoot is not able to diffuse further down. So the cells, because of the glass pieces blocking the auxin from moving further down. So that means that our shoot, cells, the shoot cells further down don't receive more auxin, higher auxin concentration, so they will not elongate more, and therefore the shoot will not bend. So which statement explains the difference between X and Y? The piece of glass destroyed the auxin. No, it did not destroy it, so it's not A, it's not B. The piece of glass in X stopped the auxin diffusing down the shaded side of the seedling. Yes, that's it. The diagram shows part of the mechanism that controls blood sugar concentration. More glucose in the blood, less glucose in the blood, more glycogen in the liver, less glycogen in the liver, more glucagon released, less glucagon released. Let's see what happens here. So a person does 1 hour of exercise, starting with the pancreas. What is the sequence of events? Which the hormone glucagon is evolved. Okay, so glucose in the blood can be absorbed by the liver and changed to glycogen, which is the stored form of glucose in the form of a macromolecule, very large molecule. So glucose to glucagon, this is caused by insulin, and glucagon does the other direction, glucagon. So that means that more glucagon is released. So we start with 5. And that means that there will be less glycogen in the liver and there will be more glucose in the blood. So 5 for 1, that is B. The diagram shows the first part of a kidney. nephron and its blood supply. During filtration, protein molecules do not pass through the wall of the glomerulus, which part contains the highest concentration of protein. So yeah, here ultrafiltration happens. A lot of things are filtered out from the blood, including a lot of its blood plasma, which is basically water. So that means, and proteins are not moving out through here. So that means if we compare C to D, it will have the same amount of protein, but because we have removed water, that means the concentration of protein is higher in D. Now we have removed solvent, basically. We didn't add solute, but we have removed solvent. Which row describes sexual reproduction? The gamete nucleus. Gametes are sex cells. They are haploid. They have half the number of chromosomes, so it's not A or B. The zygote's nucleus comes from the fusion of the two gametes, and therefore it must be diploid, and that means we have only left with answer C. Genetically different offsprings produced. Yes, sexual reproduction produces sexually genetically different offspring. Which organ secretes the most progesterone during pregnancy? During pregnancy, the placenta is responsible for producing the progesterone. Otherwise, in the menstrual cycle, it is produced by the ovaries, but at some point, the placenta. Not the placenta, but the uterus. Or, no, let me double check this. No, it is the placenta. The placenta is the organ that is grown by the embryo. So relatively early, the placenta develops in the stages of the growing embryo and fetus, and up until that point, the ovary continues to produce the progesterone, and then once the placenta is fully grown, because it's a new organ that is grown by the baby itself, this organ is capable of producing progesterone, which prevents menstruation and the abortion of the embryo. What is the role of messenger RNA, mRNA? mRNA in the protein synthesis. DNA is transcribed into mRNA, which is then translated at a ribosome into a protein. So assembles amino acids into protein molecules. This is what the ribosome is doing, moves a copy of the gene from the nucleus to the cytoplasm. Yes, so it is the copy of the gene. So the mRNA is the copy of the gene, and it moves from the nucleus into the cytoplasm to the ribosomes. The diagram shows the cell of an organism. The nucleus contains 12 chromosomes. After the cell divides by mitosis, how many chromosomes would be present in one daughter cell? 12. Mitosis doesn't change the number of chromosomes. Mitosis is for growth, repair, development, and replacement of used cells. So all cells in our body have 46 chromosomes, except the sex cells, the gametes. So therefore, by mitosis, it stays the same. A man marries a woman who has a different blood group from him. They have 2 children. The children have different blood groups from each other and different blood groups from their parents. What are the genotypes of the parents' blood groups? That must be D. This is the only option. So one of the parent has blood group AB. The other parent has blood group O. And therefore, the possible offspring will be blood group A or blood group B. So one parent is blood group AB, the other parent has blood group O, and they can have, yeah, they probably have a child with one from here with blood group A and another one from here with blood group B, all different. Pure breeding, black feathered chickens are mated with pure breeding white feathered chickens. All of the offspring in the F1 generation have black feathers and white feathers. Okay, so they are mixed black and white. This means this is not dominant recessive. Simple inheritance, this is a bit more complex. When two of the F1 generation chickens are crossed, what will be the ratio of the offspring's phenotype? So that means we have codominance here, two dominant alleles, and we can do a Punnett square again. So we have black and white. So black is this, white is this. So now we can use two different letters. So this will give us, oh, this is what we got, 100% black and white. So now we are crossing this generation again. So that means that now we are crossing black and white chickens, and what we get is BB, BW, BW, and WW, WW. That means black, white, and white. So a 1 to 2 to 1 ratio of black to black, white, and white. No, this is only because it is codominance. It's not dominant recessive inheritance here. Over the past 30 years, some antibiotics have become less effective in treating bacterial infections. What is the reason for this? That is natural selection. The bacteria evolve and over time become resistant to these. No, that is the same as what happens in nature, so natural selection. The diagram shows part of the nitrogen cycle. Which row identifies the bacteria involved in processes W, X, Y, and Z? Wow, okay, so let's start with W. Nitrate ions in the soil are changed into atmospheric nitrogen. That process is called denitrification. So, because the opposite changing nitrogen to nitrate ions is called nitrification. No, nitrogen fixation. So the Z is nitrogen fixation. W denitrification. Ammonium ions to nitrates ions is nitrification. And Y plants to ammonium ions is probably decomposition or something like this. So the organisms that do that, so denitrifying bacteria, so it's either A or B, X nitrification, yes, it must be B. Y decomposers, yes, and Z nitrogen fixation, yes. The table shows processes in the carbon cycle that release carbon dioxide into the air or remove carbon dioxide from the air. Which row is correct? Releases carbon dioxide into the air. Decay, combustion, photosynthesis, respiration. So there are many. Combustion does, respiration does, decay does as well, photosynthesis does not. So it is not C. Removes carbon dioxide from the air. That is only photosynthesis, so it must be A. And our last 4 questions. What helps to maintain fish stocks? Fishing during all seasons Probably not. You don't want to catch the young fish. Fishing in all areas of the ocean. Probably not, because then we don't have retreats for fishes. Maintaining protected areas, yes, because that gives fishes a zone to retreat to and to reproduce. Reducing the mesh size of nets, no, because then more juveniles will be caught, which causes the population to further decrease as well. The statements describe some of the events that occur during eutrophication. What is directly responsible for the increase in decomposers? So eutrophication is the influx of nutrients into a lake or a river, water environment. These increased nutrient loads are in the beginning giving plants or specifically algae more nutrients to grow. So algae will grow a lot and they will grow so much that they will cover each other, and many of the algae in the lower layers will not receive any sunlight, plus other plants that grow further below the surface will not receive any sunlight, and these organisms then start to die. That means that a lot of dead organic matter is now in the river and the lake ecosystem, which will sink to the floor, and decomposers will break down this organic matter, and doing that they will use up a lot of the oxygen in the lake or the river, because they do this by aerobic respiration, which requires oxygen. So directly responsible for the increase in decomposers is an increase in the death of producers, which follows the increase in the population of algae. There's too much competition for sunlight then, and then a lot of these algae, which are producers, algae are producers, same, will die, and other plants as well. That's why producers, not only algae, and that feeds the decomposers, which means they will increase. Which cell structure makes bacteria useful for genetic modification? That's something we need to know. These are the plasmids, the small rings of DNA in the bacteria cells. The diagram shows an industrial fermenter that is used to produce penicillin. What is the function of the part labeled X? It will stir the whole mixture so that it stays nicely mixed. So this is for maintaining an even temperature in the solution. So if the microorganisms, they do aerobic respiration as well. Aerobic respiration produces heat. So if the whole thing is not evenly mixed, then we will have some microorganisms more concentrated in some places and producing more heat, which means some places will get hotter, some will get cooler. So by keeping everything mixed, it as well maintains an even temperature. So that was the specimen paper 2 from 2023. I hope you learned something new and I could help you to prepare for your IGCSE exams in the future. Until then, have a great time and enjoy your day, and I will see you eventually for the next video and practice test. Have a good day.